Hello, everyone. Good morning, good evening to our friends across the globe, and welcome to the webinar today. First of all, I'm Sharon Kell. I'm going to be the moderator for this webinar sponsored by Awenza. I am an internist and geriatrician with an MPH in epidemiology. I was trained in the U.S. at the University of Alabama and was an assistant professor of medicine for about 7 years there, practicing medicine, teaching, doing research. Since then, I joined the pharmaceutical industry where I've been for over 23 years. I've been with initially Novartis as a scientific director in medical affairs. Then I went to J&J as director of med affairs and senior director clinical R&D. After that, Impacts Pharmaceuticals, where I was a VP of clinical R&D, medical affairs, as well as in pharmacovigilance and drug safety. And Amneal is a VP of drug safety. And while I was at Impacts and Amneal, I had the experience of managing a risk management department where we participated in a pregnancy registry. I was also at Provention Pharmaceuticals, Provention Bio, as a senior VP in clinical development. And currently I am doing consulting in those areas. So just to start off the topic for today's discussion, as you know, embarking on the journey of pregnancy, the influence of external factors like medicinal products on fetomaternal outcome is very important, has far-reaching consequences. And as we all know, during clinical trials, pregnant subjects are excluded from trials. And if pregnancy does occur during a trial, the standard procedure is to discontinue that subject from the trial and then monitor them throughout the trial and after the trial to assess pregnancy outcomes. Generally, at the time of initial marketing, there are either no data available or limited human data available about the safety of a drug or biological product taken during pregnancy. So to empower the healthcare professionals for better treatment and counseling, the FDA provides comprehensive guidelines on pregnancy registries and surveillance programs. The team at Awenza integrates their experience and clinical knowledge, FDA guidelines, and practical challenges to make this subject as clear as possible for the stakeholders. So in this webinar today, Sanjeev Meglani and Mukta Chopra, who are co-founders and directors of Awenza, will talk in detail about these challenges and solutions in the post-approval pregnancy safety studies. But however, at the onset, there's just a couple housekeeping topics. Please, and you're welcome to post your questions during the webinar. We'll keep track of them and do our best to address your questions while we're on this webinar. But in case we're not able to answer your questions due to time constraints, we will try to respond to you via email. And this webinar, the slides will be available for download on demand after a couple days. So at this time, I'm going to hand it over to Sanjeev, who's going to start today's presentation.
Sure. Thanks, Sharon, for moderating this session. Hi, good morning, good evening, everyone. My name is Sanjeev Meglani, and I'm MD in internal medicine. Well, both Mukta and I started Awenza Life Sciences 4 years back, and at Awenza, we are doing end-to-end pharmacovigilance and clinical safety for our clients across the globe. Before starting Awenza, I was the Vice President of Pharmacovigilance at APSA, where I was responsible for their pharmacovigilance and medical affairs for their US operations. And prior to that, I worked in Accenture as their Vice President for Pharmacovigilance and Medical Writing for their US and European clients. And before that, I also worked as a medical director for a global CRO called CliniRx. And prior to joining the pharma industry, I worked as a senior resident in the field of cardiology and medicine. Yeah, so Today, in our presentation, I'm going to talk about the pregnancy registry. And in this presentation, well, I'll give the brief background initially. I'll talk about the advantages and limitations of the pregnancy registries. I'll also talk about the protocol design, including the objectives, the strategies for the statistical analysis, how the data is collected, how the data is analyzed. And then my colleague Mugdha will talk about the factors which will influence the protocol design, including recruitment or retention. She'll also talk about multi-drug pregnancy registries, and she'll also talk about the role of scientific advisory board. Well, I think first of all, a very important thing is what exactly is a pregnancy registry. So a pregnancy exposure registry is practically an observational prospective cohort of women receiving a biopharmaceutical product of interest as a part of their routine clinical care who are enrolled on a voluntary basis during gestation before the outcomes can be known. So as a part of this pregnancy registry, basically the pregnant women are allowed followed up until the end of pregnancy, and in fact, like even after the birth of the child, like up till 1 year or even sometime even longer. So the data is basically collected, and the intent is to collect the pregnancy outcome, and if there are any specific congenital malformations or any particular birth defect that are detected, either during the course of pregnancy or even after the birth of the child, so they are collected. Well, if we look at the FDA data and the US stats, pregnant women represent a very important segment of the population with over 6 million pregnancies occurring per year, which is based on the national vital statistics of the US. Pregnant women may have chronic conditions. These could be like diabetes or epilepsy or asthma that need to be treated during pregnancy, or the pregnant women may develop acute or serious medical conditions during pregnancy that require treatment. And in nearly half of these pregnancies in the United States may be unintended, and these can result in potential inadvertent exposure to drugs and the biologic products in pregnancy if a woman is exposed to a drug when she's not aware that she's pregnant. So it's very, very important, imperative to for the safety information on the to get that safety information on product exposure during pregnancy. So practically during the clinical trials of the drugs or the biologic products, the pregnant females are actively excluded from the trials. And in case if a patient becomes pregnant during the course of a trial, so the standard process is to discontinue the treatment and monitor that patient to assess the pregnancy outcome. So consequently, at the time of a drug or a biologic product's initial marketing, except for the drugs and the biologic products developed to treat conditions unique to pregnancy, practically there are no or limited safety human data. And therefore, it's very important that these pregnancy registries are conducted, and we can get the adverse outcomes during the time of pregnancy or even post delivery. Well, if we look from the FDA perspective, like the Food and Drug Cosmetic Act, so it authorizes FDA to require certain post-marketing studies or clinical trials for prescription drugs which are approved under Federal Food and Drug Cosmetic Act and the biologic products approved under the section of the Public Health Service Act. So under this particular section 50503, FDA can require such studies or the trials at the time of approval to assess a known serious risk which is related to the use of the drug, to assess a signal of a serious risk which is related to the use of drug, or identify an unexpected serious risk when available data indicates the potential for a serious risk. So practically, in terms of pregnancy registries, advantages and limitations, we all can understand that there is a huge advantage of pregnancy registry in collecting the data. But then at the same time, the pregnancy registries does have some limitations, and therefore there are some other means also to collect the data. So I'll first talk about the significant advantages of pregnancy registries. So the pregnancy registries are practically often the initial proactive step in assessing the safety of use during pregnancy of new drugs after they're first marketed, of course, because they provide a number of advantages over other approaches. While many pregnant women use medications, the use of individual drugs can be quite rare. So by enrolling an exposure group made up only of women who took the medication of interest, so these kind of pregnancy registries are quite helpful and efficient for evaluating the effects of infrequently used drugs in the population. So this is practically a distinct advantage over other study designs such as case control studies and the small healthcare utilization databases, which practically do not have sufficient power to evaluate the outcomes following rare exposure. The second important part of the pregnancy registry, which is a significant advantage, is that the longitudinal nature of the pregnancy registry, they allow the estimation of the absolute risk of the pregnancy outcome. For example, registries that enroll women before any prenatal testing has been performed can estimate the risk of malformation amongst infants whose mother used a drug of interest. So this is basically in contrast to a case control studies that estimate risk related to that for a reference group. Another important advantage is that the pregnancy registries can compare the risk of outcomes amongst women who have used a variety of treatments for a single condition, including different monotherapies or different polytherapy combinations, or practically no treatment or at all. So an additional advantage is that a single registry can monitor a variety of pregnancy and infant outcomes after after medication exposure, including the postnatal outcomes. Now, while there are a lot of advantages of pregnancy registry, there are some limitations. And while we understand that these registries are often very helpful or quite efficient means to assess rare exposure, they sometimes practically lack the statistical power to evaluate the rare outcomes. So most teratogenic exposures do not increase the prevalence of all malformation, but they would have more selective effect on the individual defects or distinct patterns of defects. So the pregnancy registries are powered to detect the common outcomes such as the total prevalence of all malformation, and they can detect only very large increase in these rare individual defects or the patterns. However, many drugs associated with adverse effects in pregnancy result in only moderate increases in these rare outcomes. So therefore, the pregnancy registries are quite limited in their ability to detect teratogenic effects on specific malformation with statistical certainty. So another important limitation is the lack of a comparable reference group at times. So ideally, like in a pregnancy safety study, a comparison group should be drawn from the same population as women with the exposure of interest, using the same method for recruitment, same method for enrollment, and ascertainment of the outcome, so that both the groups practically have the same baseline risk for the adverse pregnancy outcome. Another important thing that we have to see is that the finding from a pregnancy registry may not be generalized to the broader population of the women who use a drug. So enrollment of women in pregnancy registries typically is voluntary and self-selected, and registry participants represent a very small proportion of all the women who have taken a drug. So for these reasons, the characteristics and the experience of women who participate in a registry may differ from those of the known participants, and these characteristics may modify the effect of a drug. Well, it's very important that in this field of drug safety, especially for the pregnant patients, we adopt the good PV practices, and FDA guidance document clearly lays down a lot of emphasis on the good PV practices. And the first and foremost thing is that the good PV practice involves a collection of comprehensive data on the adverse pregnancy outcomes to detect the signals and develop a case series for analysis. So there can be different kind of sources, and the sources could be spontaneous reports which are submitted to the sponsor and FDA, as well as there could be case reports from the literature, from the clinical trials. So well documented and informative case reports can be used to identify a signal, particularly if the pregnancy outcome is rare in the absence of drug exposure. The safety signals, they generally indicate the need for further investigation, which may or may not lead to the conclusion that the product caused the outcome or increased the risk of the outcome. It's very important that the quality of the data or the report that we are collecting is up to the mark, because it is critical for appropriate evaluation of the potential association between the drug and the adverse events. So FDA recommends that we should be doing a very diligent follow-up. In fact, like whenever the pregnancy registries are conducted, or for that matter, any pregnancy safety studies are conducted, so there should be a proper milestone or time points when the follow-ups have to be done, like say at the end of first trimester or second trimester, or at the end of third trimester, or at the time of delivery, I mean. And then even after birth, it can be done 1 month or 6 months or 12 months after the birth of the baby. And there should be standard data points which should be collected. So the case reports are the most important source of reports of adverse pregnancy outcomes, but often it can be challenging to interpret because the information is incomplete. So we should try to get follow-up information as detailed as possible so that we can get good case reports. And a good case report should practically have a detailed description of the adverse pregnancy outcome. A good case report should have a detailed description of the exposure, including the specific medication, the frequency, the route of administration, and the duration. A good case report should have information regarding the timing of the exposure in relation to the gestational age. It should have detailed information about the maternal age, medical and pregnancy history, any kind of exposure to the known or suspected environmental teratogen. So all of these things are very important, and it's important that the case reports contain all that information. Well, there are some disadvantages also of the case reports, and that's why there is a role of these pregnancy registries and the observation studies. So the known limitations of a spontaneous post-marketing reports, they pose considerable challenges in analyzing the cases and determining whether a causal association exists between the drug and the adverse event. So therefore, the routine pharmacovigilance generally would be insufficient for a conclusive assessment regarding the potential risk of an exposure during pregnancy because of the inability to quantify risk. And that's why like these observation studies or the pregnancy registries or the pharmacoepidemiological studies, they are practically needed to provide additional information, including a control group, to derive and compare rates on the safety outcomes of the drug and the biologic products used during pregnancy. Well, a sponsor, as per the FDA guidance, should have a structured approach for any pregnancy surveillance with the targeted questionnaire to obtain follow-up information. So practically, like say at the end of the first trimester, there should be standard questions like what was the report for ultrasonography or amniocentesis, or if there was any particular specific lab investigation that was done, or if there was any congenital malformation that was picked up maybe in the second trimester or third trimester. So the questions that are designed should be appropriately documenting the outcomes. It should basically have all the parameters, as a result of which the analysis can be done later when we get the data for these pregnant women. Well, a well-written protocol for a pregnancy registry should practically describe its objective, and which may range from the open-ended surveillance to testing a specific hypothesis. So if we look at the FDA guidance, it talks about the objectives, like what practically should be there as a part of the objective of pregnancy registry. And the overall purpose of the pregnancy exposure registry, as we all know, is to provide the human data on the safety of the biopharmaceutical products during used during pregnancy. And typically, a pregnancy registry should have a specific primary and secondary objectives, and they should be defined very clearly. So most of the pregnancy registries would have their primary objective as to assess the risk of MCMs, or practically, which is major congenital malformation, in the offsprings of women exposed to a given drug just before or during the pregnancy. And the registries can evaluate multiple maternal, obstetrical, fetal, and infant outcomes, from pregnancy complications to development delays. Moreover, they may provide an opportunity to evaluate not only the safety, but sometimes also the effectiveness of the drugs, as well as the risk which could be associated with untreated diseases during pregnancy. So practically, the pregnancy registries can also evaluate the effects of dose and the gestational timing of exposure, as well as the effect modification by the maternal characteristics. Well, in terms of the study population for inclusion, ideally, women in the exposed and the unexposed cohort should be enrolled in a pregnancy registry prospectively. That is, before the conduct of any prenatal test that could provide knowledge of the outcome of pregnancy. If the condition of the fetus has already been assessed through prenatal testing, say some ultrasonography has been done, or say amniocentesis has been done. So these kind of reports traditionally have been considered retrospective. However, because it may be difficult to obtain enrollment before prenatal testing on a consistent basis, the study population should include all women, including those who have had early prenatal testing. And the protocol should practically address how pregnancies with the prenatal testing before enrollment will be evaluated in the statistical analysis to avoid potential bias. Well, it's very important that the outcome definition and ascertainment is clearly documented in the protocol. And again, whenever any such protocol is submitted to FDA, they're very particular about these kind of things. Like it's a very clear mandate as per their guidance document that these outcomes should be very clearly documented. A pregnancy can result in live birth, miscarriage, which is loss before 20 weeks, elective termination, or fetal death or stillbirth, which is lost after 20 weeks. So within each of these categories, the fetus or infant can be evaluated for the presence or absence of the primary outcome. So as a part of the study design, so the protocol should state the criteria for defining the study outcomes, and criteria for defining birth defects as major should be clearly stated. So what exactly is a major congenital malformation? According to FDA guidance, the major congenital malformation might be defined as abnormalities in the structural development that are medically or cosmetically significant, which are present at birth, and they persist in postnatal life until or unless repaired. So similarly, the criteria should be established for abnormalities that will be excluded from the definition of outcome, which could be, say, they are minor or transient. There could be genetic syndromes, positional defects, or prematurity related defects. So a standard classification should be used as appropriate. Another important thing I would like to emphasize here is that the FDA typically expects that even though there are different milestones where the healthcare professionals or the pediatrician after basically delivery, they would be kind of getting in touch with the mother to get the information, or during the time of pregnancy, the obstetrician will be consulting with the patient to get the additional information as a part of the registry at times. But then if there are any malformations that are identified for those particular scenarios, an expert clinical geneticist or a dysmorphologist should review and classify the medical records. So it's very important that a clinical dysmorphologist also review and classify medical reports, of course, if there is a major congenital malformation, but not on a routine basis. Some examples of other outcomes that may be primary or secondary on a case-to-case basis could be like measures of fetal growth deficiency, the preterm delivery. There could be other pregnancy complications, development milestones, or neurological abnormalities in the offsprings of exposed mothers, and there could be abnormalities of the immune system development in the offspring of the exposed mothers. Well, as a part of a well-structured protocol, it is expected that there is a clear-cut documentation laid down for the sample size and the statistical power. And a protocol for pregnancy registry should include a statistical analysis plan with a description of the target sample size based on the power calculations and the assessment of feasibility of the study in the patient population of interest. So whenever we are estimating the target sample size, it's important to take into account the expected background rate of the pregnancy loss and the cases that may be lost to follow-up or otherwise unevaluable. So estimated risk based on the general population may not apply to specific disease groups like, say, diabetes or multiple sclerosis. Determination of an adequate sample size depends upon the objectives and the design of the registry and the background rate of the outcome in the study population. So if we are talking about more than one pregnancy outcome, which we are taking into account, so the sample size should be based on the outcome with the lowest background rate. And emphasis should be given to the prevalence of the disease in females of reproductive potential and the pregnant women, and anticipated frequency of the product exposure in the pregnant women. So practically, I think in the pregnancy safety studies, we all understand that no known teratogen increases the risk of all major congenital malformations. So typically, as I mentioned earlier, a specific defect or pattern of defect is associated with a specific teratogenic exposure during a critical period. So the specific major congenital malformations occur rarely in the general population. It could be like fewer than 1 in 10,000 or fewer than 1 in 1,000 live births. So historically, the pregnancy registries have not had sufficient sample size or power to evaluate the increased risk for specific major congenital malformation unless the relative risks are large. And therefore, most of the registries, the pregnancy registries, compare the overall proportion of the total combined number of various MCMs or the major congenital malformations observed in the exposed group to the overall proportion in the comparator groups. Now, there can be situations when pregnancy registry is not feasible. So in those situations, the important question is how is the safety evaluation carried out? So if the pregnancy registry is not having adequate power to allow statistical achievement, so and that can also happen whenever the exposure to pregnancy is quite low, and it happens like for some indication there is a possibility that the exposure is quite high, and there will be more cases of pregnancy or the pregnant female patients for a particular study. But sometimes the achievement of an adequate sample size may not occur when the likelihood of the exposure in pregnancy is low, or sometimes we know that the use of product is not recommended during pregnancy, so obviously the exposure would be quite low in such studies. So anticipated issues with the registry study feasibility should be stated in the protocol and appropriately at least. For example, by expanding the inclusion criteria to include all reports of exposed pregnancies, and that can include both prospective and retrospective. So for the products which are anticipated to be used rarely during pregnancy, like say in the treatment of advanced cancer indication, so sponsors can indicate a pregnancy surveillance program, which is basically a structured approach for data collection with targeted questionnaire to obtain follow-up information on all the exposed pregnancies of which sponsor becomes aware. And this type of case series of exposed pregnancies can inform clinical and regulatory decision making. And worldwide safety collection data is usually needed to identify a sufficient number of exposed pregnancies for a clinical safety assessment. Well, a very important piece of information that should be adequately described in the protocol, and it's a very big challenge also in the pregnancy safety studies, is the selection of the comparator group or the reference group, because it's not always possible to have a comparator group in the pregnancy registry. So the most valid reference group will have a comparable outcome definition and outcome assessment. And ideally, each registry is basically constructed to include one or more internal reference group. Whenever this is not possible, an external reference group must be selected with care, and each comparison group has its own advantages and disadvantages. So if, for example, an external population-based reference group is generally larger and can provide more stable estimates for the specific malformation, while if you talk about an internal comparison group, it may be too small to support assessment of specific malformations, and may be able to provide more comparable estimates for malformations overall. So more than one comparison group can be used to increase the study generalizability. And in anticipation of the possible conflicting or confounding factors, however, a primary comparison group or the group should be identified, and it should be justified in the protocol. External comparators are usually considered less valid than the internal comparator. For example, if we talk about a randomized controlled trial or an observation study on the relative risk of stroke in the elderly population exposed to a drug, it would never use an external reference. However, the pregnancy registries often compare their estimates with the background risk in the standard populations. So external reference groups must be used with caution, since the estimated risk of major malformations can vary widely depending on the population, the definition, and ascertainment methodology. So while when external references are the only alternative, they must be appropriate to the population being studied, and analysis should at least take into account the characteristics of the surveillance program and use the same methodology in their exposed group. Well, it's very important that as we are talking about the internal or the external comparator group, or we're talking about the sample size, the exposure definition and ascertainment is also uh, clearly documented. And if we are concerned about the structural malformation, the etiologically relevant period of the exposure is practically the first trimester of the pregnancy. And to identify this period, we need to establish the gestational timing. So the obstetricians typically time the pregnancy from the first day of the last menstrual period, determined by maternal recall, or preferentially it could be by uh, early pregnancy ultrasound. And depending on the pharmacological characteristics of the drug, the specific defect and the accuracy of the timing information, the window of interest might be the second or the third month. So anytime during the first 4 months after the LMP, or even weeks before LMPs for those kind of drugs which have a long half-life or unspecified period of effect, for example, in the cases of vaccines. So exposure later in pregnancy can affect other outcomes. Well, the treatment strategies change during pregnancy and the doses are commonly adjusted. So it's recommended that the detailed information is collected on the start and the stop date, the frequency, duration, and the indication. In terms of exposure ascertainment, a question is like how registries collect the information can affect the accuracy of the data. So some kind of registries obtain information from the women themselves, and women generally know more about their habits, the occupation, the medical and the ops history, and the compliance with the drug than the individual health care provider. So the other registries rely on reporting by the clinicians and have no contact with the patient. So clinicians can provide more complete and accurate information regarding diagnosis and indication. However, there is a risk of exposure misclassification if women stop or incompletely comply with the prescription drug regimen during pregnancy. So a woman might be more willing to tell an interviewer not related to her care than to tell her doctor that she decided to reduce her dose or quit taking a medication. Yeah. So the other point is identification of covariates, and the sponsor should consider the potential for confounding by indication, which makes it difficult to determine whether any observed effects are caused by the drug or biologic products or the underlying disease. And in terms of covariate, the data should be collected on the data on pregnant women's pertinent like medical history or current disease status and the overall management. And then there are other confounding factors also which have to be taken into account, like any data on the socioeconomic status, the maternal age, the tobacco and alcohol use, illegal drug use. So all of these factors have to be taken into account as other potential confounders in the covariates. I think another important thing is the data collection, which is obviously the basic foundation for any pregnancy registry. And it's important that the data is collected appropriately. Like any other epidemiology studies, registries can kind of benefit from the technological advances in communications, that is their ability to enroll subjects through social networks and collect the data through the confidential websites. So, and in fact, like whenever we are preparing any protocol for any pregnancy registry, there will be certain recommendations, in fact, which are already laid down in the FDA website. And first and the foremost thing is that FDA in fact encourages that the sponsor should have a kind of a website which should be dedicated to the pregnancy registry for the product, and the patient should be able to enroll on these websites themselves. Also, of course, there can be multiple options. The patients can talk to the pregnancy registry rep by calling at the toll-free line. So on the prescribing information or on the website, the toll-free number should be displayed so that the patient or the healthcare professional who wants to enroll a patient they should be able to talk to the pregnancy registry rep at the dedicated toll-free line number, and then the data can be collected, of course, through the questionnaire. And it's very important that there are different means to recruit the patients so that the data can be collected appropriately. And of course, Mugdha will be talking about recruitment and retention aspects of the pregnancy registry. So the pregnant women are ideally enrolled into the pregnancy registry before or soon after the exposure of the interest, and then followed throughout the pregnancy. Well, if a live infant is born, the infant may be followed for a period typically ranging from 3 to 12 months, and data may be collected at several time points. It could be collected at the enrollment. Information may be collected on the product exposure, maternal characteristics, prenatal testing, and other baseline data. Data may be collected during the second or third trimester of pregnancy. A brief update on the status of pregnancy and exposures can be obtained at that point in time. Again, at the end of the pregnancy, information may be collected on additional exposures during pregnancy, risk factors, and details regarding the pregnancy outcomes, including any congenital anomalies, gestational age, and birth weight, and perinatal complications. For live births, data may be collected at several points in time. Typically, it can be at 3 months, 6 months, or 12 months on the infant characteristics and healthy outcomes, including the congenital anomalies or defect or malformations, or any other outcomes pertinent for the drug of interest. And data can be collected as a part of a targeted follow-up, and that may be needed to collect additional details on the specific outcome of the interest. So the source of the baseline and the follow-up data is an important consideration that needs to be taken into account, and it's best to evaluate all options for obtaining data to determine the most appropriate source for the specific data requested. And the pregnant women can provide detailed data on the drug use, including the exposure to the drugs of interest as well as other drugs, on the relevant risk factors, and on the pregnancy outcome. Now, while we are talking about the data collection, it's also important that data analysis and presentation is also done appropriately. So the validation of the cases should be performed through the medical record review, and adjudication of the outcomes, as I mentioned earlier, it should be done by a clinical dysmorphologist in case there are major congenital malformation, and it should be done for both the exposed group and the comparable group. And practically, like inferential statistics should be applied to the test pre-specified hypothesis regarding the potential association between the drug and the between the drug exposure, and the outcome of interest, which could be any specific defect if there is any. So potential bias should be discussed as a part of the data analysis, as well as the possible methods for mitigation if applicable. So descriptive statistics are primary approach for summarizing patient characteristics and additional data from a pregnancy registry. And practically, given the heterogeneous nature of the data obtained in pregnancy registry, there is practically one single format for data presentation that is applicable for all study. Like there will be scenarios where the exposure could be practically very low. So in those kind of situations, what we have seen is that only the descriptive data is provided, because the data or the patient exposure is so low that the other statistical methodologies may not be practically applicable. So at the end of the day, based on the type of the nature of the drug and the exposure, how much exposure we are expecting, so the data analysis and the presentation is done. So at this point in time, I would like to now hand over to Dr. Mugdha, who will be talking about the different aspects of pregnancy registries. She'll be talking about the factors affecting design and success. She'll talk about the multi-drug registry and also the role of scientific advisory board. And probably before I hand over to Mugdha, just a brief introduction I'd like to give for Dr. Mugdha. So she is also the co-founder and director of Evinsa Life Sciences, and she has over 16 years of experience, including pharmacovigilance and clinical research. Before starting Evinsa, she was Associate Vice President for US Pharmacovigilance and Clinical Safety at APSA, and she was overseeing delivery and operations from India, which included but not limited to case processing, aggregate reporting, RMPs, literature monitoring, and signal detection. And before joining APSA, she was with Ranbaxy as a manager in the Department of Medical Affairs and Pharmacovigilance. And she comes with an extensive experience in various safety database like Argus or RSG for case processing, reporting, and product license management. and she's also met significant challenges of business continuity planning and data restoration. Over to Mugdha.
Yeah, thank you so much, Sanjeev, for talking in detail and for the nice introduction and giving in details in your presentation about the pregnancy studies, their objectives, collection and analysis of data in the pregnancy studies, and assessment of safety of the products by analyzing the outcomes. We know that exclusion of pregnant and lactating women from pre-approval clinical trial results in lack of safety and efficacy information for their population, and it necessitates the post-approval research. So observational safety studies of pregnant women and their infants are imperative to ascertain the impact of the product exposure and assess the risk of the adverse pregnancy or the fetal outcomes. The pregnancy studies complement the pre-approval clinical trial data and add to the evidence regarding the product safety and effectiveness. The draft guidance released by the FDA in May 2019 for pregnancy safety studies increased the rigor of pregnancy safety studies considerably. And FDA recommends the sponsors to conduct these studies more and more frequently. So there has been an upward trend in the number of pregnancy safety studies being conducted by the pharmaceutical companies after the release of the guidance in May 2019. The approach and design of the pregnancy study encourages participation and facilitates data collection, resulting in greater sample sizes and thorough assessment of the safety. And one of the most important parameters to be considered for inclusion in the design of the pregnancy safety study is the recruitment and the retention. So successful recruitment and retention strategies are very critical to the success of the pregnancy studies, such as registries or other studies requiring enrollment of study subjects. And FDA recommends a robust recruitment and retention plan that includes a multi-pronged approach to ensure widespread coverage of the eligible population. Early enrollment also may improve detection of the pregnancy outcomes such as miscarriage. And these plans should be flexible and continuously reassessed throughout the study to ensure that the registry maintains an adequate number of eligible pregnant women in both the exposure and the comparator groups. One of the greatest challenges with pregnancy studies is recruiting eligible patients, and appropriate recruitment strategies should be adopted for recruiting the eligible patients. So engaging healthcare providers and patients before the initiation of recruitment increases awareness of the study and provides an opportunity to seek feedback from these stakeholders regarding the study plan. And sponsors should collaborate with entities such as existing registries, patient advocacy groups, medical societies, and other relevant organizations to engage in the awareness activities. Under the pregnancy and lactation labeling rule requirements, if there is a pregnancy registry for the product, relevant contact information must be included in the product labeling under the subheading of pregnancy exposure registry. Suggested modes of contact information include a toll-free telephone number or a website's URL, basically Uniform Resource Locator. So creation of a targeted and customized awareness plan for the study assists in reaching healthcare professionals and patients in a variety of settings. To encourage enrollment, it is always helpful to create a registry website and to place the links to the website on other prominent websites like FDA's list of pregnancy registries and sponsors and product specific websites. So FDA has a list of pregnancy registry at their website, and all the pharmaceutical companies who are actually conducting the pregnancy registries or pregnancy safety studies, FDA encourages them to submit this registry information at their website, and the links to these registries are available at the FDA website, so that it creates more awareness to the patient population, to the pregnant women, to the healthcare providers, to everybody, so that there is more recruitment and retention in these pregnancy safety studies. FDA provides a huge support in recruitment for these pregnancy studies. The FDA's Office of Women's Health, OWH, maintains an online list of pregnancy registries that are actively enrolling women to raise awareness about pregnancy registries and connect consumers and healthcare professionals to the registries. The registries are posted to the FDA's OWH webpage, based on the sponsors or investigators' request to list its registry. But FDA encourages sponsors and investigators, as I was saying, to submit a pregnancy registry listing to OWH at their dedicated email ID, which is registries@fda.hhs.gov. And FDA at the same time does not endorse any registry and is not responsible for any content on the registries listed on the web page. These are all the responsibilities of the pharmaceutical organizations or the investigators who are actually conducting the registry, but FDA encourages them to list the registries on the web page, but they are not responsible for content or mentioned in the registries which are mentioned in their links. So recruitment strategies can be described as facility-based, healthcare provider-initiated, or patient-initiated. Facility-based recruitment can occur at the level of a practice or health system. Electronic health records can be used to identify drug or biological product users to facilitate the enrollment process for providers. For example, an automated alert of a pregnancy registry can be generated in response to positive pregnancy test results and the specific drug or biological product prescriptions. With regard to the patient-initiated recruitment efforts, they rely on patients to contact the registry study staff and self-enroll themselves. Because pregnancy is often recognized by the patient first, so registries that enroll patients directly can allow for recruitment of the patients earlier in pregnancy. So it is a very important parameter for the recruitment of based on the patient-initiated strategies. So useful avenues to notify the pregnant women of pregnancy registries, they include various avenues like print media, including publication, press releases, articles in newspapers and magazines with pregnant women among their readership. And there can be other avenues like distribution of flyers and posters in locations such as hospitals, ultrasound clinics, laboratories, prenatal classes, community centers, stores, and coffee shops. And the most important avenue is social media, we all know, which will help in notifying the pregnant women for the pregnancy registries. And last but not the least, the downloadable applications for mobile devices or personal computers, which could enable broader participation through ease of providing the information to the pregnant patients. Healthcare provider initiated recruitment of patients is an important deciding factor for many pregnant women, and provider recruitment approaches, they include announcement of registry study and contact information in the product labeling, as we were discussing earlier, that the product labeling should have the website information or a toll-free number so that the healthcare provider can contact or can just register the patients on the pregnancy registry, some promotional materials and the product internet pages, announcements in professional journals and newspapers, personal mailings to the specialist, and presentation and exhibits at the professional meetings. So successful strategies to engage the participation of pregnant women in the safety studies could include the incentives that facilitate study participation. So that can also include employing empathetic, culturally sensitive, and personable study staff, basically. So which means that if the study staff is able to link themselves or able to have that empathy with the patient, so then it becomes a more an important point for the recruitment strategy, and more pregnant women can be recruited early in the pregnancy stages of the pregnancy registry. Even though recruitment materials may yield strong initial recruitment results, FDA recommends implementing a robust retention plan to ensure that an adequate number of pregnant women remain in the registry for a longer period of time. The retention plan should address specifics of patient retention strategies, contingency plans to obtain follow-up information, methods to track follow-up rates over time, and implementation steps to improve follow-up if expected follow-up rates are not being met. So retention plan is a very, very important parameter in the pregnancy safety studies. So we should have a proper, robust retention plan documented in the protocol as well. FDA also recommends that retention efforts focus on participating healthcare providers to improve retention rates and reduce the burden of data collection. like implementing streamlined processes and succinct forms for the collection of the data. Access to pregnancy registry results basically provides a strong incentive for the participation of healthcare providers, particularly obstetric care providers, and the provision of interim data reports to participating healthcare providers, which can also increase the retention. Additionally, high levels of retention have been achieved by pregnancy registries that communicate directly with the patients, emphasizing the mission of the pregnancy registry, which can reinforce participants' motivation to remain in the study. Sharing study results through a newsletter or a website, which has been found to be more effective in reinforcing patients' reasons for participation and being retained in the study for a longer duration of time. Again, establishing and maintaining a longitudinal relationship between participant and the interviewer can also reduce the losses to the follow-ups. As with the other longitudinal studies, collecting contact information of family members or friends in case the patient cannot be reached, it can aid in retention. Recruitment and retention of pregnant women may be aided by a flexible follow-up schedule, like conducting follow-up interviews by telephone during evening or the weekend hours, or over a secure online platform, because participants may be, balancing the work and the childcare responsibilities. So flexible follow-up schedule has to be must in and should be enumerated in the protocol as well. Thus, it is very critical to design flexible models that encourage patient enrollment. Solutions, some of the solutions include models like in-person and remote enrollment, as well as flexible recruitment methods that utilize advertising and social media. Streamlined data collection also relieves the burden on the patient and the healthcare provider. For example, allowing data collection from home and without any undue burden at the healthcare facility. Again, patient reimbursement, which is also a key, which can also help to increase the recruitment and the retention. To prevent overburdening patients, physicians, and healthcare delivery systems with multiple requests to participate in individual study, FDA encourages sponsors to work together directly or through consortiums to develop or support multi-product registries. A multi-product pregnancy registry actively collects information on exposure to various product therapies in specific diseases, such as HIV or epilepsy or other diseases. And in some cases, a general multi-product registry, such as that conducted by a teratogen information service, collects information on products for unrelated indications as well. Multi-product registries have advantages over the single product registries with respect to the efficiency and economy. And they also have a specific advantage of having comparison groups of pregnant women unexposed to the drug or the biological product of interest, which is readily available. Another very important piece of information is till when the pregnancy registry be continued, or when should pregnancy registry be discontinued. Pregnancy registry should be continued until sufficient information has accumulated to meet the scientific objectives of the registry. It should be continued till the feasibility of collecting sufficient information decreases to unacceptable levels because of very low exposure rates, because of poor enrollment, or because of losses to the follow-ups. And any other method of gathering appropriate information becomes achievable or are deemed preferable. Another very important aspect is lactation study added to a pregnancy registry. There is often a need to collect lactation data to provide information on the safety of the drugs and the biological products during breastfeeding. Pregnancy registries can be used to recruit and enroll breastfeeding women in lactation studies, because some women enrolled in a pregnancy registry are already taking a drug or a biological product during pregnancy, and because they may be likely to continue the treatment after delivery. So these women are the ideal population in which to study the product levels in the milk. And there is a separate guidance document which is also published by FDA for consideration of study designs for the lactation study. When we talk about the factors affecting the success of any pregnancy safety study, it is very, very important for all of us to understand that the privacy of the subject data is very critical, and privacy of in fact, the subjects, the personal data of the subjects is very important in these kind of pregnancy studies. So sponsors should consider privacy, including data protection, and human subject protection, including obtaining informed consent and the IRB oversight when designing a pregnancy registry and developing protocols for the subsequent use of data from the registry. FDA strongly recommends that an IRB should be consulted when developing a pregnancy registry to ensure that the collection of data and all other procedures associated with the registry will withstand scientific and ethical scrutiny. Because pregnancy registries typically do not involve the administration of an investigational drug, there is not likely to be any foreseeable risk or harm to the pregnant women or to the fetus or to the resulting child from participating in the registry, other than the risk associated with inappropriate disclosure of the identifiable private information. The patient should be requested to sign medical record release forms to allow collection of the records from the healthcare providers of the mother and the infant. Investigators are responsible for ensuring that any data releases, they are compliant with the Health Insurance Portability and Accountability Act (HIPAA) in the US, and obviously GDPR in the Europe, and that all research performed complies the standards of privacy of individually identifiable health information. If the registry involves the collection of information on child after birth, so there is generally obviously the follow-ups which are conducted during each trimester at the time of the delivery, and obviously once the child is born, so follow-ups are generally conducted till 1 year after the birth of the child. If the follow-ups are conducted either through a physical examination or a specimen collection, so the consideration should be given to 21 CFR Part 50, which is additional safeguards for children in clinical investigations for privacy of the human subjects' data. Now, to ensure the scientific integrity and appropriate patient protection, it is advised that each registry should have an independent data monitoring committee or scientific advisory board, similar to those which are used in the clinical studies. Members of the committee could include experts in obstetrics, embryology, teratology, pharmacology, epidemiology, pediatrics, clinical genetics, and any relevant therapeutic areas. The committee could assist in review of the data, classification of specific pregnancy outcomes, including the major congenital malformations, miscarriages whenever relevant, and the dissemination of the information to ensure that results are interpreted and reported accurately. The role and duties of the committee or the scientific advisory board should be specified in the protocol clearly. So at the end, I would just like to say that appropriate and flexible study designs and protocols are imperative for assessing the product safety during pregnancy. And appropriate analysis of multiple sources of data provides a holistic view of the product safety in pregnant women and her infant and the journey through pregnancy, delivery, and beyond. So this is all what I have in my presentation today. So I would like to open the floor for the questions.
Yeah, thank you, Magda. We do have a few questions. The first one will go to Sanjeev. The first question is there a standard process for the subjects to get registered in a pregnancy registry?
Sure, Sharon. So I'll answer this. Yeah, so practically whenever we are preparing a protocol for any pregnancy safety study or registry study, it has to be defined very clearly how the patients can get registered. So there are multiple methods. One could be a simple method wherein if a sponsor is having a particular pregnancy dedicated website, so the patients can enroll directly on the website, and then obviously like the system is configured in a manner that the email alert gets notified to the concerned pregnancy registry rep, and that person can then contact the pregnant patient to obtain the follow-up or get additional information. Otherwise, obviously, it is always encouraged that the toll-free number of the pregnancy registry is mentioned on the website. Of course, the patient can directly approach the pregnancy registry rep by calling at the toll-free line, or they can kind of communicate with that person, and then of course the patient, the pregnancy registry rep can coordinate with the patient and get the additional information. Even the registration can be done through healthcare professionals also. So the treating gynecologist, they can obviously enroll the patient and then they can let the sponsor know. So all of these things have to be defined in the protocol. So the patient, while they're going to the clinic and when they inform that they become pregnant while they've been taking some drug. So if the healthcare professional is already aware of that program, so even through that HCP, the patient can be registered. And in fact, on the website, sometimes there is a dedicated page for the ICP and then for the patient. So even the ICP can enroll the patient on their behalf.
Thank you, Sanjeev. Second question, what is the role of complementary studies?
Yeah, so practically, as I was mentioning earlier, the pregnancy registry studies sometimes have limitations because they sometimes lack the statistical power to kind of figure out the rare exposures. So sometimes there are some additional data points, like there could be post-marketing safety information coming from the interim reports, or there could be some spontaneous reports or case series from which a particular safety signal may become apparent, which may not be becoming apparent from the routine pregnancy registry study. So these additional data points practically form a kind of complementary study. So FDA guidance document does define about the role of the complementary studies, and it could be like electronic data sources like the insurance claims and the electronic health record databases, or the population-based case control studies wherein there is some data. So practically the idea of complementary studies is to complement the routine ongoing pregnancy registry, so that if a particular pregnancy registry is not getting enough data in terms of the rare outcome, at least the data additionally can be obtained from these extra sources, and some signal can be derived in total.
So we are out of time, but there's just a couple more questions left. Should we go ahead with these?
Sure. I think, yeah, we can take one or two more.
Okay, so Magda, I think this should, you could address this one. How long are pregnancy registry studies conducted?
Sure, Sharon. I'll address this question. So basically, the pregnancy registries have to be continued till as much time as possible, because we all know that the exposure to the pregnant women is minimal, and the patients, there are not many patients who get enrolled in the pregnancy registry. So to get enough data for assessment of the outcomes, for assessment of any kind of miscarriages or major congenital malformations. So we have to have enough data with us to evaluate, and for that data to appropriately assess the safety, thorough safety assessment. So we have to have enough information about all this, about all the data which we are collecting, and so the information should be collected across a long period of time. And in fact, FDA nowadays is giving the post-marketing commitments to the pharmaceutical companies to conduct the studies, longer pregnancy studies, maybe for 7 years or maybe even for 9 years or more, just to have enough data associated with the assessment, for the proper assessment of the outcomes.
Okay, thank you, Magda. The last question for Sanjeev, how are major and minor malformations differentiated?
Sure, Sharon. I can answer this question. So yeah, in fact, FDA guidance document talks about the major malformations and the minor malformations as well at the same time. So practically, the major malformations are the kind of defects which could be major structural defects, which could be defined as abnormalities in the structural development that are medically or cosmetically significant, and they are present at birth, and they persist in postnatal life unless or until repaired. But then also, whenever the protocol is being prepared, so the criteria should be defined for a major or minor, because sometimes there could be some defects which may seem to be major, but then they might not be actually major. Say, for example, there could be a ventricoseptal defect, which is a small muscular ventricoseptal defect, which we know may spontaneously close with no consequences. So the criteria should already be defined. So such a kind of a small VSD would not be considered major. Similarly, like when we talk about minor, as far as the definition goes, the minor malformations are basically those uncommon structural differences in the infant that have no serious medical or cosmetic consequences. And again, the criteria can be defined very clearly, because sometimes there can be some defects which can be clearly ruled out. Like say, similarly, the positional deformities are often excluded. Say somebody has the infant has abnormal head shape, but that practically spontaneously resolves shortly after birth. So they're not basically considered to be a malformation because they spontaneously resolve after birth.
Okay, well, thanks, Sanjeev. Thanks everybody for your participation. So I think that ends our webinar. Have a good day. Thanks.
Thanks everyone. Thanks, Sharon.
Thank you
Thanks,
so
Mumtaz.
much,
Bye.
everyone. Thank you. Have a nice weekend. Thank you, everyone. Bye.